Hello everyone, my name is Dongwan Hong. I'm happy to present my research material, and in addition, I'm first visiting to Singapore. Is very nice building and good city, and yesterday Professor Zhang's talk is very impressive to me, so everything is wonderful. The title is an implementation of anti-cancer drug miner using 3D protein structure prediction. However, some change the title because I will introduce the history of my research area and two ongoing projects: AI study using multi-omics data. Let's see the contents first. Review and reminder of AI study using multi-omics data. As we know, multi-omics data is very heterogeneous and different data type and big size. So we find and decide to the category of research goal and according to the target. Because, for example, disease and patient categorization and functional and biological study and treatment patients are not different suitable data formats. So. Next, ongoing project using AlphaFold 2. AlphaFold 2, a very popular, fantastic tool to predict the 3D structure of mutation and certain genome. So first, we'll introduce an infectivity prediction of COVID-19 mutation, and finally, cancer drug repurposing using AI. It's very similar to previous talk. Yeah. Maybe 10 slide. After 10 slide, you used to introduce the AI study using omics data to our students in our lab. So first, disease and patient categorization. You should adapt the data, gene expression, and DNA methylation. For example, as you know, the cancer type, cancer grade is the fixed standardization defined by WHO, ACMG, and so on. So stage 0, 1, 2, 3, and 4. So, however, this study there are 17 histological types, and after the diagnosis, using the methylation data of CCNS, and it improved the treatment score. Functional and biological study is we should recommend to the DNA sequence. The popular journal is DeepSeq. DeepSeq using genomic sequence and 1,000 base pairs chromatin organization. This is the DNA sequence and predict and the in silico function. So using after got the results, so we use the CRISPR-Cas9 and the DNA editing and adapter and so on. However, this is n-scale samples. Many samples, many data need to is a very difficult thing. Finally, treat a patient in our hospital. Wants to predict the side effect and the drug response, and many guys wants to the precise result. For example, clinical cancer research 2018, they used the autoencoder and using the data mRNA, microRNA sequence as well as the methylation data. This data is adapted to real hospital. They find and get the precise, accurate result of the side effect and cancer prognosis. The first case study and prediction of drug response using the gene expression, clinical information, and somatic mutation. As you know, there are different data types. So different data type means the decrease the accuracy of predictions. Many guys pilot study try to adapt this model using more therapy response using omics data. So our laboratory try to simulation the predict the drug response and the lung cancer and bladder cancer of TCGA. So we train the data is having the response or non-responder data of platinum therapy. We want to, unfortunately, we get the AUC score is, as you see, the 0.619. So we failed to the suitable data, so discarded to submit or any journals. However, 1 year later, some guys published the National Review of Clinical Oncology in 2018 and very similar scores. At this point is the prediction using multi-omics data wants to get the accurate or precise result. 1 or 2 weeks ago, I tried to summarize the PubMed data, the keyword of the cancer and machine learning and deep learning and so on. So, So deep learning didn't get the precise high score on AUC. However, many machine learning will get the good score more than deep learning data, deep learning model. So I guess that this is the very different data type, big size. So we first we understand the meaning of the data and classify the data and reduce the dimension. And many things are required to increase the accuracy. First, systematic study design according to complex and heterogeneous data types. After that, selection of training model. The means is the supervised and reinforcement learning, and so on. Data notation is a very big problems. For example, IL-2, IL-2, interleukin-2, and metadata and draw is not finalized in standardization. So it's needed to correct the data schemes. Finally, error correction, misclassification, for example, responder, non-responder, switch, the misclassification. So, and finally, for removal familiar errors, A-oxy and transversion, paraffin block problem, germline contamination will correct the fix and these problems after finally we want get the around about the 0.9 accuracy. First, I introduced the application of proteins 3D structure prediction. First, a prediction of an infectivity of SARS-CoV-2 mutation. So this news introduced the last year. I'm impressed the title, the entire protein universe. As we know, AlphaFold DeepMind introduced the 200 million proteins in harboring and stored in database. As you know, who subject to I study, our laboratory, they're hiring the bioinformatician or computational biologist. Bioinformatics in a point is about engineering. They have to develop an algorithm and database and tools. Position computational biologist in the final discover the new unknown factor or novel discoveries. It's a good point of discovery. So our laboratory seems like to feel like the gold rush. We can, what can we do for medical research? First, we decide to develop some algorithm or system to human genome is very big size to adapt to the AI. So the COVID genome is 13,000 base pairs, so very small compared to the human genome. So adaptable use the AlphaFold 2. So AlphaFold 2, Dr. Professor Chang introduced yesterday, so it's very good scores at these days. And another, so this line means in spite of the endemic, however, these lines reproduce the SARS-CoV mutation regeneration again and again. So the PNAS tell about some virus, Lassa and Seoul virus is very risky and infections in the world. So we gather the database and the related to COVID-19 data and classify the genome and protein data, epidemiological data, and drug and target data. So we join the data, so distributed site and some more get novel ideas and novel interesting. So this finding is good to me. Amino acid properties result in SARS-CoV pandemic. So sorry. These regions are RBM of the SARS-CoV spike protein. So color, red, sky blue, red, sky blue, red, sky blue. This position is the very infection site. Among the polar, switch the polar to non-polar, and among them, so after that, K and R, change the amino acid K and R, a very high infection score. So, according to the accumulation of a big and huge amount of data, we find the point. So we want to evaluate the score and model and SARS-CoV variant. So first, SCPS, this is the SARS-CoV mutation predicted on AlphaFold 2. After then, clustering. Clustering 1 and 3 is very high infectivity score, and as I told you, polarity change score. Polarity change score is the polar and switch non-polar is high score. So they field are independent values. So developed evaluation score APSS. APSS score in the significant regions, we know the high infections VOC risk. So we predicted the novel unknown mutations using the machine learning, and some mutations are predicted and novel and increase the infections, and down decrease the infection. We finally, using in vitro experiment, we validated our prediction and the in vitro result experiment. It's almost the same, similarity. Yeah. So we developed the IVE system because many medical researchers and biologists are difficult to use use and manage the GPU and gather the amino acid mutations and calculate. This is very difficult. So we use to calculate and management data is on IVE system. One other topic, however, I don't have enough time, so drug repositioning. So result. I will show the result. the CRPC patient, they have not proper treatment. They many patients will die. We maybe the 10% of CRPC patients, the novel gene fusions, many TMPRSS2 ERG fusions are well discovered all prostate cancers. However, the CRPC does not have enzymatic activity. So we on other fusions, novel fusions, they have enzymatic activity. So we find the wild type, mutation, fusion, they have structural changes, and V600E wild type mutation and fusion have same structure and same proteins. So we in vitro validation P3 means the harboring these fusions and the prostate cancer cell line. We find the mutation, fusion have to decrease the cancer toxicity, and we will prepare to make the organoid. So it prepared to the in vivo experiment. You're using method escape. Finally, target therapy drugs built up as just 3 inhibitors. In spite of that, EGFR mutation, many inhibitors. That means there is the clinicians, for BRAF inhibitors, easy to pass the IRB or Korean FDA. So try to the clinical trials of all inhibitors. You don't need to use your system. However, EGFR has many inhibitors. If you use the score and grade, getting to the results, it will very provide the benefits of the cancer treatment. So we'll try to the imaging and whole genome analysis and the integration analysis will support to decide in cancer, our hospitals. So, yeah. Thanks for your attention. I'm sorry over time. Yeah.
Thank you. Thank you for the talk. And any question for Dr. Hong's talk? Yeah, you can go ahead.
Thank you for the presentation. Yeah, very impressive. I just have some question about that treatment response prediction part that you mentioned that someone has successfully improved the AUC from 0.6 to 0.9. That's really impressive. But however, could you please provide more details about what exactly he did? Because this part about misclassification. However, that as far as I understand, that responder and non-responder are defined in a certain way, and this should not be replaced with each other, even though there should be a vague area. So could you please explain it a little bit more? Thanks.
So look at your AUC figure.
Yeah, ASP go. Not have ASP go. So I don't have any figures in AUC. However, I recommend to you first reduce the data, because multi-omics data have the sparseness. Sparseness means I recommend you the negative selection, not positive selection, negative selection. That means choose the best data set, make the best data sets. After then, so we select the best training model, some expression, mRNA and protein expression data. So
Yeah.
good scores the supervised reinforcement learning, and after they're discarding the errors and they'll get the AUC scores. Yeah, is it okay? Yep.
I actually understand that one should do data dimension reduction and, for example, use lasso or any similar techniques. But for this error correction part, I really don't understand why you should, because you just said misclassification here, right?
Oh.
And I don't, could you please, because perhaps I missed the detail, could you please just elaborate it a little bit more why that can be exchanged or something?
This is the very good position, good point. In my experience and worked on hospital, so the typing, for example, typing means the HPV positive, HPV negative, and responder or non-responder has the human error or misclassification. They be almost about the 20%, about 20% if they're correct and fix the error, so we get there some good scores.
Thank you. I think I have this similar thinking about this, like the proportion of each class. Yeah, thanks.
Yeah, thank you very much. Yep.
So next.
Thank you for the nice talk. I want to ask, like, for the mutation prediction, what features did you use? And are there some amino acid specific features that you used for mutation prediction in SARS-CoV-2 infectivity?
Please repeat slowly one more time.
So for the mutation prediction,
Mutation prediction in
what
COVID
is the
mutation?
feature you are using for training
In
your
COVID
model?
mutation?
Yes. Features, what are the features used?
The feature you train your model, what kind of feature you are used for training your model here?
First time we try to deep learning model, for example, LSTM. So we don't make sense of understand the result exactly. Why the result will come to us? Not predict or not measure the mutations. At this stage, I know the deep learning have to get the well-structured, modified data in predict the SARS-CoV mutation. So we will very small phone, random forest, ensemble, RGBoost has the same similar scores than prediction in the unknown mutations.
Yeah,
Is it okay?
Yeah.
Yeah.
For this non-polar to polar mutations,
Pardon?
non-polar to polar mutations,
Non-polar.
Non-polar to polar mutation.
yeah.
Polar. As we know, amino acid is the polarity picture. So non-polar, polar is these amino acids are including. So we just the gathering and integrated WHO GSAID, and we find the polar and non-polar changes that occur to the just on the infection. We want to correlate the risk factor. However, it's not just only infectivity. Yeah.
Okay. Thank you so much.
Yeah. Thank you.
John?
Just a quick question. Under the one where you're doing the error reduction and you said you're going to remove the 8-oxo-G part of it, I'm a bit troubled by that because it's an important cancer pathway, and particularly in certain cancers more than others. So I'm just a bit like, why did you decide to do that, and how are you going to exclude the true, where 8-oxo-G is very implicated for colorectal, for lung. This is an important pathway, so I just wanted to hear.
It's a good question. It's a good question. This question is the very long time ago with deputy the ICGC Peacock and TCGA. So I'm working at the ICC Argo mutational signature working group, and we together work the Arvin. Where's Arvin? And so OXOG is the first proposed founded in the neuroblaster is the high sonication, the Illumina machine, because of the high sonication plus the OXOG. OXOG remove the discarding OXOG to discard OXOG is I don't know the software, the name of the software. So
Okay. It's okay. We can talk
sorry
offline.
about that,
We can talk offline.
Yeah.
I think. Thanks.
yeah, thank you.